Today we'll be talking about genomes. All right, so here I've included a snippet on the Human Genome Project. In short, the goal of the Human Genome Project was to determine the sequence of the human genome. So determining the nucleotide base sequences and location for every gene in the human genome. So it was started in the 80s by the Department of Energy and then eventually taken over by the National Institute of Health, or the NIH. And then in early 2000s, it was eventually completed and published by Nature magazine, or Nature journal. All right, so some of the methods that were used or in the Human Genome Sequencing Project was PCR, shotgun sequencing, electrophoresis, and Sanger sequencing, which I've included a video on the newer method of Sanger sequencing, which uses capillary electrophoresis. So to kind of overview shotgun sequencing is where you actually take, many of the large pieces of DNA that you want to sequence, and you cut them into randomized fragments. So you cut this DNA into randomized fragments, and what you do is you line up the DNA with other DNA to find its relative location in the overall sequence. So we could take this magenta one, it starts with this GCT and ends with GTTA. Well, let's say you find another sequence, another piece that starts off with GTTA. Well, you would know that from the GTTA up until the CATA would be another fragment. So you would actually be able to move from the GTTA to CAGT, GCATG, and then you would find another sequence like the purple guy here, and then line it up, find that overlap, and then you're able to eventually create a seamless sequence using all of these little fragments. So that's shotgun sequencing. All right, so Sanger sequencing is going to be a way of actually finding the sequence in order to splice together the sequences through shotgun sequencing. So Sanger method is going to actually give us that sequence that we're looking for in order to line up the overlapping parts of DNA. So first we're going to denature our double-stranded DNA using heat. And what we're going to do is we're going to make multiple copies of a segment. And making multiple copies of a segment could be done through PCR, for example. Or what you could do is you could take that unknown segment of DNA and you could splice that into, let's say, for example, like E. coli, which is a type of bacteria. You could splice it into the genome of that E. coli. And what you could do is take that E. coli and allow it to replicate and replicate. And every time it replicates, along with replicating its own genome, it's actually replicating the fragment of DNA of interest that we've spliced into its genome. And then next, we create a primer of some sort, and that primer could be, let's say we're using the E. coli example. The E. coli's genome would be known, and what we could do is create a primer that matches the E. coli, or is the anticodon for that segment of, or a segment of E. coli DNA, which eventually carries on and leads into that eukaryotic piece of the genome that we were trying to code for. And then what you do after you've created your primers is you have, let's just say, 4 independent trials that we're going to be running. So we have four different, you could just say vials, that we're going to be running experiments in. So in this purple one, what you're going to have are all of the nucleotides necessary for carrying out DNA replication, or you could say, all of the substrate needed for DNA polymerase will be in here. However, 90% of all of the A in here, so you're going to have adenine, you're going to have thymine, you're going to have cytosine, you're going to have guanine, all in here. This is all the substrate needed to, follow through with DNA replication. Now, 10% of all of that adenine is going to be a modified, or you could say marked, form of adenine, where actually a second oxygen is removed from the backbone, and it's actually called dideoxy ribonucleotide. So dideoxyribonucleotide. And it's also marked in a way that we would be able to detect it. So 10% of all of the adenine in here is going to be a marker, you could say. And the special characteristic about that marker is because of its structure, as DNA polymerase moves through and is creating new DNA using a template strand, if you integrate one of those marker nucleotides, what will happen is your DNA polymerase will stop. It'll stop there and it'll disband and it'll pick up another piece of DNA somewhere else and then continue with that. Now the same thing goes for, thymine. 10% of all of the thymine in here is going to be marker thymine. 10% of all of the cytosine in here is going to be marker cytosine, and then 10% of all of the guanine in here is going to be marker guanine. And then what you do is you throw DNA polymerase into there and you let it kind of run its course. Many pieces of, or not many pieces, many of that enzyme, many DNA polymerases, many strands of that DNA, and then many nucleotides. So you have all the substrate you need. However, what's going to happen is, we could just start off with this purple, DNA polymerase will synthesize DNA, synthesize DNA, synthesize DNA, synthesize DNA, until it eventually reaches a place where it integrates, has a 10% chance. Every time it reaches a place where it would normally integrate an adenine, instead of integrating a regular adenine, it might integrate a marked adenine. And when it integrates that marked adenine, it dislodges, leaving the small fragment of DNA. Now the same thing's going on multiple times, many times, in all of these vials. And what you're doing is you're creating many, not necessarily random, but you're creating many sequences of DNA of various lengths. But all of the, you could say all of the ones in this purple vial are going to end in adenine, because the only thing that's causing it to stop is the marker adenine, right? So all of the fragments, large or small or medium, are all going to end in adenine. And then what you do, if you remember from 160, we learned about gel electrophoresis. What gel electrophoresis does is first off, the gel is porous, and what we do is we inject into wells in that gel DNA. And that DNA, we run a current through the gel, and that allows DNA to pass from one side to another side. And the thing is, that gel is going to be porous, meaning things can pass through it. However, things are going to pass through it at different rates. So your big things are going to not travel as far, and your large things are going to travel very far down the gel within the same period of time. And so we could actually start off with this yellow guy here. Your shortest fragment is going to travel the furthest, and it's going to end in T. So you have this short fragment that traveled the furthest, and it ends in T. Now let's look at our second shortest fragment, or the one that traveled the second furthest. That one ends in C. And then what about our third shortest fragment? That one ends in G. And so from here you can kind of infer, your sequence is probably going to go T, C, G, A, T, G, and so on. A way to simplify this is, this was the electrophoresis sample that I gave, or a electrophoresis sample that I gave, or that I could give. And if you were to compile all of this data together into a single lane in that gel, this is what it would look like, each color denoting a different letter of the alphabet. So that's, in short, Sanger method, or Sanger sequencing. All right, so back to the eukaryotic genome. Humans have 3.2 billion base pairs in their haploid cells, or base pairs in their haploid genome, you could say. So 3.2 billion base pairs in 1N um, genomes. And the human genome has about 20,000 protein coding genes. Now, the average gene is about 27,000 base pairs, and your genes can be about 1,000 to 2.4 million base pairs long. So if we look at bacteria, for example, they're, anywhere between, like 10 to the 5th power to about 10 to the 7th power in terms of the amount of nucleotides they'll have in their genome, nucleotide pairs in their genome, and their haplogenome. And then when we look at, our eukaryotic types, they're a little bit larger in their genome size. And if we look at humans, they're kind of between, 10 to the 9th and 10 to the 10th over here. Now, the thing is, humans, for example, have way more DNA than bacteria. However, your bacteria are actually going to be composed, or at least their genome is going to be composed mostly of coding DNA, whereas a lot of our eukaryotes their genomes are going to consist mostly of non-coding DNA. So we have a similar number of genes as other organisms, yet we have more DNA. We have more DNA volume in our genome than other organisms. So therefore, humans will have more non-coding regions. So if we look here, our humans will have 3.2 billion base pairs in our haploid genome. However, we're only coding for about 25,000 genes. Now, if you look at a mouse, they have 2.6 billion base pairs in their haploid genome, and they're coding for about the same amount of genes as a human. So we have more non-coding regions of our DNA. So 99.5% of our genome is identical from one human to the next. We have 3.3 million single nucleotide polymorphisms, or you could say, mutations that have carried on to just be considered variations in a gene. So 33.3 million single nucleotide polymorphisms, or some people will call them SNPs, which account for only 1/5 of that 0.5% variation. So a single nucleotide polymorphism, that's just going to be, an alteration in a nucleotide, which in a nucleotide within a gene that would normally code for a protein, when you eventually get into, the transcription and translation, the sequence of amino acids might be different. And if the sequence of amino acids in a gene is different, well, you might actually get a different phenotype, for example. So these are just going to be variations in our genes. So only 1/5 of that 0.5% variation is due to single nucleotide polymorphisms. Now, half of our genes only have one copy in the genome. However, the other half have multiple copies in the genome. So we're going to all have different copies of different genes within our genome. And that's going to account for 4/5 of that 0.5% variation between humans. Now we also have these things called pseudogenes, and those are going to be inexact, usually non-functional copies of genes that are oftentimes going to be located near the same gene, and often it's a result from the accumulation of mutations within a gene whose product is not required for the survival of the organism. And so that's going to only contribute to a small fraction of that 0.5% variation. So most of it's going to be due to multiple copies of a single gene, of any of our genes within our genome. All right. So protein coding genes only account for 1.5%, around 1.5% of our genome. Now, we consider this 1.5 or the other amount of our genome, so the 98.5% of our genome. Some people will call it junk DNA, which I wouldn't necessarily consider junk DNA because they actually do have function, but 98.5% of our genome does not code for genes. or genes that would eventually be expressed in the form of proteins. So these regions that are non-coding, they have function, and so they are going to be able to contribute to DNA structure and regulation. So things like promoters or promoter sequences that'll recruit all of the enzymes associated with transcription. Enhancers, which again is going to help with transcription. So regulation, things that are going to play a role in regulating gene expression. We also have parts of our DNA that's responsible for coding for ribosomal RNA. And we know that our ribosomes aren't necessarily, used in, they're not decoded, they're not translated. Our ribosomes are actually a structural component of gene expression. And we also have these other things called microRNA, which are stretches of DNA that will code for pieces of RNA, and the function of microRNA is actually to bind to and potentially silence mRNA that could maybe be translated. So it'll actually create microRNA with the anticodon sequence for some of the genes that we're trying to express. It's kind of like a self-regulation process that microRNA has. And then we also have large stretches of repetitive DNA sequences. So we have highly repetitive sequences, and we also have moderately repetitive sequences. So our highly repetitive sequences, which are going to typically be, less than 100 base pairs, we call those satellites, and they are typically repeated thousands of times. So thousands of repeats. Highly repetitive, have thousands of repeats. And these regions are typically not going to be transcribed, so they're going to be a part of things like our centromeres, or they could be a part of these things called short tandem repeats, which are sequences that we could use for DNA fingerprinting, for example. So if you remember from 160, you might have had a crime scene lab where restriction enzymes were used. Essentially what happens is these things called restriction enzymes have a tendency to cut your DNA at specific locations throughout, the various loci, or they will cut your DNA at specific loci. And the thing is, let's say, you want it to cut DNA here and here. Well, if you have a large amount of short tandem repeats in between them, you're going to have a very large chunk of DNA, and like maybe two small chunks of DNA at the ends. But if you have a very short amount of short tandem repeats in between those two cut sites, you're going to have a very small piece in the middle and maybe very large pieces on the outside. And so, in short, what happens is, you could go to a crime scene, take a blood sample, and expose it to restriction enzymes that are going to cut your DNA at very specific locations. And then what you'll get after running a gel electrophoresis are going to be, you're going to find these bands that travel at different distances. And as we know, when we're running gel electrophoresis, the smaller pieces travel further and the larger pieces don't travel as far. And that's all going to correspond with the size of the chunks of DNA. And the thing is, if you have a large amount of short tandem repeats, or a small or a medium amount of short tandem repeats, the number of short tandem repeats that you have in between each cut site is going to determine the distance that it travels through the gel electrophoresis. And so through doing that, we're able to kind of create a genetic or DNA fingerprint for any individual. And so what you would do is you would take suspects' blood, you'd take samples of the DNA from their blood, expose those to restriction enzymes, and match up fingerprints from suspects to the crime scene, for example. So here's a little picture depicting that. You have your DNA sample, you extract DNA, you use restriction enzymes to cut the DNA at very specific locations. You run it through electrophoresis. The longer strands stay towards the beginning, and the short DNA fragments travel further, and then you end up with a DNA fingerprint. All right, so those are going to be your highly repetitive sequences. What about your moderately repetitive sequences? These are going to be repeated from 10 to 1,000 times, And normally these are going to be transcribed, but not necessarily translated all the time. So things like your tRNA or rRNA, which again, uh, these are going to be a part of the machinery in gene expression, but not necessarily translated. We know that tRNA has the function of carrying amino acids to the ribosomes for translation, and we know that rRNA is going to be the substance that composes the majority of our ribosomes, which again, machinery, not necessarily coding. So transcribed but not translated. So these are going to be code sequences that are transcribed but not translated. Um, and then we have these things called transposons. So these are kind of interesting, and I'll kind of be short with this one. So these are going to be sequences that will move around the genome. Some people consider them to be cellular parasites, and so let's say this is a transposon. Uh, this gene is actually going to code for an enzyme called transposase, and so the function of transposase is actually to come around and extract your transposon gene and splice it into another part of your genome. So it could actually disrupt the expression of certain genes, but it's self-fulfilling because it codes for the enzyme that allows it to propagate. Very interesting. Potentially long topic if you wanted to talk about it. All right, and then just to kind of like recap everything or to see the bigger picture here. So your single nucleotide polymorphisms could actually be used to identify disease and risk for disease. So we could actually run genetic testing to kind of determine a person's propensity to develop a disease, for example. So like breast cancer, there's a single nucleotide polymorphism found on chromosome 8 that if they are a heterozygote for that single nucleotide polymorphism, they have a 20% chance higher of getting breast cancer, versus if they are a homozygote, meaning two of the same alleles, two of those same single nucleotide polymorphisms, they're at 63% higher chance of developing breast cancer. And so pharmacogenomics is going to reveal how some single nucleotide polymorphisms could increase the likelihood of drug effectiveness depending on your genotype, or you could say single nucleotide polymorphism for a specific receptor for a drug, or a specific protein that interacts with the drug, for example.